Chemistry is universal. Molecules are not mere bystanders in the grand tapestry of cosmic change. They evolve alongside and help shape, sometimes dominate, the process of forming nebulae, stars, and planets. In the laboratory on Earth, we can study the individual molecules directly in front of us. But in the vast reaches of space, we must rely on the signals imprinted by molecules on the electromagnetic spectrum through their light-matter interactions to identify, quantify, and explore their chemistry. The study of molecules in space is called astrochemistry, and spectroscopy is the tool with which we conduct our investigations. Spectroscopy is the study of the interaction of light with matter, in our case, molecules freely floating in the void of space. Because of quantum mechanics, this interaction is unique to each chemical species. The wavelengths or colors of the light the molecules interacts with, either by absorbing that light or emitting it, form a pattern that is distinct to that molecule, a different fingerprint for each and every molecule in the universe. This makes spectroscopy one of the most powerful tools available to humanity to learn about the chemical compositions of matter. Indeed, spectroscopy is used every day to study our atmosphere, perform medical tests, explore the properties and functions of cells, test the purity and safety of our water, food, and drugs, explore our universe, and much more. The widespread use of this tool in our society is due in no small part to Dr. Beckman's pioneering efforts. It was once an expensive and time-consuming process to build and maintain specialized equipment to measure spectra of nearly any sort. Through the invention of his DU spectrophotometer, Dr. Beckman brought an affordable, easy-to-use instrument to market that would allow not only scientists and engineers to study spectroscopy, but made the practice widely accessible to industry and the public sectors. Today, researchers still build powerful, new, more specialized spectrometers in the laboratory suited to their specific problems, but many of these are then picked up, refined, and simplified by commercial interests to make them widely available, following in Dr. Beckman's legacy. My group is particularly interested in using rotational spectroscopy, the study of the patterns of light given off by molecules as they tumble freely end over end in space, to identify complex carbon-bearing molecules in interstellar space and understand how they influence the chemical inventories that will eventually seed planets. Through the support of the Beckman Young Investigator Award, we have been building rotational spectrometers to study molecules that may be the interstellar building blocks of life-giving organic compounds, as well as a particular class of species called polycyclic aromatic hydrocarbons, or PAHs. On Earth, PAHs, which consist of numbers of fused rings of typically 5 or 6 atoms, are a large component of the soot remaining from incomplete fossil fuel combustion, as well as the char marks on grilled foods. In space, as much as 25% of all interstellar carbon is thought to be locked up in PAH molecules. As astrochemists, we study these molecules, both prebiotic species and PAHs, by first measuring their rotational spectra using our instruments here on Earth, and then using radio telescopes to search for those same spectral patterns in the regions of space where stars and planets will eventually form. Last year, we used one such instrument in our laboratory built by a postdoc, supported by the Beckman Young Investigator Award, Dr. Gabby Wenzel, to study a molecule called 2-methoxyethanol. This molecule contains many of the most important ingredients for life: carbon, hydrogen, and oxygen. Its presence in space was predicted based on chemical intuition, but its spectrum had never before been measured. Using the data collected from our instruments, combined with data from colleagues in Florida and France, my graduate student Zach Fried was not only able to measure and catalog its rotational spectrum, but to subsequently detect the molecule in space for the first time. Its abundance relative to other species in the massive star-forming region NGC 6334I, located in the Cat's Paw Nebula, is providing new insight into the interactions of prebiotic molecules with the intense ultraviolet radiation given off from the forming protostars. More recently, Dr. Wenzel and others in our group have constructed a new state-of-the-art microwave spectrometer specifically designed to study aromatic molecules like PAHs. Astrochemists have known with near certainty these molecules existed in space since the mid-1980s by looking for their spectra in the infrared region of the spectrum, were signatures of their vibrational motion, the stretching and bending of the atoms with respect to each other through chemical bonds, rather than their rotational motion, or rise. While also unique, these signatures are often extremely similar to one another, making disentangling the individual types of aromatic molecules from the infrared data so far impossible. As a result, our knowledge of individual aromatic molecules in space was extremely limited. This changed in 2018 with the observation of the aromatic molecule benzonitrile, which contains a single six-membered ring, followed by the detections of 1 and 2 cyanonaphthalein, PAHs with 2 fused six-membered rings, in 2021. These discoveries were made using the rotational spectra of these molecules, rather than their infrared vibrational spectra. Since then, using the spectrometer built with the support of the Beckman Foundation, as well as instruments from our colleague Dr. Michael McCarthy at the Center for Astrophysics, Harvard and Smithsonian, our team and his has measured the spectra of dozens of cyclic, aromatic, and polycyclic aromatic molecules, an astonishing number of which we then immediately detected in space. Combined with additional detections from colleagues in Spain, led by Dr. Pepe Chernacharo, the curtain on PAH chemistry is finally beginning to lift, providing an unprecedented view into the life cycle of carbon in the cosmos. Not only are we detecting individual PAHs in space for the first time since their presence was inferred from that infrared data in the 1980s, but the place we're finding them, a cold, dark molecular cloud where a star hasn't even formed yet, suggests they are a fundamental part of the natal chemical inventory of planetary systems from the very beginning. Indeed, the chemical archaeological record of our own solar system, as evidenced by samples recently returned and analyzed from asteroids like Ryugu and Bennu, are showing many of the same PAH molecules we're finding in interstellar space, further supporting the idea that these species are critical players in the delivery of organic carbon to planetary systems. The instruments we're constructing in the laboratory to continue to push the frontiers of interstellar chemical complexity are highly specialized, built on techniques invented only in the last 15 years or so. But companies like BrightSpec are already working rapidly to follow Dr. Beckman's legacy through simplification and generalization of the technique. Soon, like the DU spectrophotometer, these too may see widespread use in industry ranging from pharmaceutical development to commercial manufacturing environments. All the while, we will continue to use them to pursue our understanding of our cosmic chemical origins.